And on average, you get 1, 2, 3, 4, 5, 6 thin filaments around it. So again, those cross bridges are reaching out in all directions to connect with the thin filaments. Okay, molecular composition. So we're talking about thick filaments. So basically, if we were to tease apart the myosin, right, you have these heads kind of going all around to connect with the actin. If you were to tease one of these heads out, you see that you have the heads with a binding site where ATP joins up, and you have the tail. So it's kind of like, it's like that paddle is continuous with me, and it's not that I'm a human. It's like I'm some kind of weird snake creature because I have a tail that goes all the way down past that wall that anchors in and intertwines with all the other tails. So now, it's not just that we're a human international rowing team, but we're freaks of nature. Like gamma radiation got to us, so we're all a bunch of snakes that have our tails all intertwined. It's kind of like weird. They're all offset and staggered, and the thing is you need ATP in order for the site to bind. So what happens is ATP comes along, and the head gets cocked in the head, and then ATP becomes ADP and PI, and then it joins with the actin and PI leaves. It does the stroke, the ADP leaves, and then in order for the myosin to disengage from the actin, you need another ATP. So think about this. You need ATP for the myosin to disengage. What happens if you can't supply ATP? Well, what happens to the myosin? It's stuck there. So when is an extreme situation where you would have myosin head stuck there?
Rigor mortis.
Rigor mortis, which is when so you really don't care because you're dead. That's why when people die, they're like, for a while, until the proteins start to degrade, because there's no ATP generation to pull that myosin head off of the actin. The things you learn.
So, like, what happens when, like, let's say you get a really bad leg cramp?
You cry. You like roll around. They're not exactly sure. There's two theories. One is electrolyte imbalance. The other is that I think it's neurological fatigue. But they're not 100% sure. I mean, the thing you should do is if you get a really bad, say, calf cramp, is to stretch your calf. And it should alleviate the cramp. It should solve the cramp, which seems counterintuitive because you don't want to stretch it. But if you stretch it, it should solve the cramp right away. But they're not 100% sure. We don't know. So maybe, Carly, you could go do your PhD in that and tell us all. And come back in 7 years and tell us. Be like, what did you find out? And you're going to be like, I found out that PhD is really expensive, and this is why you cramp. So there you go. That's a thick filament. The thin filament is known as myosin. And like I said, I mean, you're thinking right now, myosin thick, actin thin, but oh no, we have two other proteins called troponin and tropomyosin. Sorry, so I have to ask at this point, have you already learned about musculoskeletal contraction within physiology? Yeah? Okay. So this is kind of review.
Yeah, this is like,
Okay, well, he's exposed you to that information. Okay, you've been exposed to the information. I'm not going to test you on the steps involved in musculoskeletal contraction, but you should learn it because chances are you'll probably get quizzed on it in physiology. You will be tested on it in exercise physiology. What I will say is you should be familiar with the four major players. The four major players for shizzle, right? Because there's big dog myosin. Like, I'm big dog myosin, don't be messing with me. And then there's little dog actin, is not as big as myosin, but he's like, hey, little dog actin. And then he's got his two little brothers, troponin and tropomyosin. So troponin and tropomyosin, you need to be familiar with what they are and what they do, but I'm not going to ask you for specific sequential steps in all of it. But they're all involved in the contraction site. But basically, on actin, you have these active attachment sites for the myosin. Right? And the myosin will connect with the actin and be like, hey, little bro, give me a little pound. Right? And then they need a Red Bull to disconnect, or the ATP to disconnect. They're very close. Very, very close. sarcoplasmic reticulum and the T tubules, you have this amazing network that was designed or you've been involved with, or maybe like some aliens came along and planted us here, whatever your view of how we got here is. Sarcoplasmic reticulum is basically, it's these tubules that sit around the myofibril. They run along the long axis of it, and basically they store and then release the calcium, because it's the release of the calcium which gets the troponin and tropomyosin out of the way so you can get the contraction occurring. The T tubules is basically it's basically going into the cell, and it increases the muscle cell's fiber surface area, and this basically it allows for the most uniform contraction of the muscle possible. Because if you didn't have the T tubules, there would be a delay, and then instead of being like the beach is that way, it would be the beach is that way, right? Because it wouldn't be exactly uniform contraction. So it'd be kind of, you know, funky, right? Look like you just got tased, bro. So they penetrate the interior cell, and it forms a nice little triad within there. So I mean, really what you need to be familiar with is sarcoplasmic reticulum is what stores and releases calcium, and the T tubules are what help with the kind of simultaneous transmission of the nerve signal to all the areas of the muscle fiber. Hey Slater, how's your baseball team going? I'm good. I hate to tell you, Jays are out. There is a triad relationship. So the T tubules contain this membrane protein that go into the intramembrane space. And sarcoplasmic reticulum is commonly referred to as SR. And they also go into the space, but the whole point of this all being is on page 287, you have a really nice diagram of how the sarcoplasmic reticulum and the tubules of it and the T tubule basically just infiltrate each sarcolemma so that you really have very efficient contraction of the muscle fibers at once. Because if you only had to say around the individual muscle cell, all the peripheral muscle fibers would contract first, and then the interior muscle fibers would contract. But because you have this system, this triad, not triads like, you know, Japanese dealing drugs and, you know, killing each other, but triad in terms of, you know, the gang. Triad in terms of this infiltration from the sarcoplasmic reticulum and the T tubules, all of the myofibrils will be contracting pretty much at the same time. There will be a slight delay, but pretty much be contracting at the same time. Let's see, the sliding filament model of contraction, so you learned about this in physiology, if not, or sorry, you've been exposed to this information in physiology. No problem. So contraction is basically the cross bridges form, so those paddles go out, touch the actin, move the myosin closer, release with ATP, and cause contraction and muscle fibers contract. And then contraction ends when the cross bridges become inactive. Now we're not going to talk about the whole series of muscular contraction because I'm assuming that's going to be covered in physiology, and then also in exercise physiology that will be covered a bit also next semester. What else? In the relaxed state, the thin and thick filaments only overlap a little bit, and then when you get the sliding filament model contraction, during contraction the thin filaments slide past the thick filaments. So it's not so much that me being myosin, I'm pulling myself forward, but it's really that I'm pushing the desks back towards the other set of actin over there. Or in this case, we'd be pushing this actin towards that actin, and that actin would be coming in, so that with the muscular contraction, what we're getting is basically, okay, I'll wheel up for a second, is basically we get contraction, relaxation, except it's you myosin there, and me myosin here. Come on, myosin. And we do and relaxation, right? Now everybody's going to be like doing this motion with desks during their exam. So when the nervous system signal comes along, the myosin heads are permitted to bind from the cross bridge, and then that contraction begins. Cross-bridge attachments form and break several times. So now imagine that instead of, say, me and Paige pushing it this way, what we're doing actually is we connect, push, and all these desks are connected. Imagine it's one long table, and then we connect, push, connect, push, right? Right now, the second floor, are like, what in the world is going on? The Z discs are pulled towards the M line, the I band shortens, the Z discs become closer, the H zones disappear, and the bands move closer to each other. So basically what happens is, you have your H zone, you have your Z line, right, you have your H zone, so you have the actin, you have the myosin here, so the H zone is from end of actin to end of actin, it's only myosin. The Z line is the anchor point, you got the M line, which is the anchor point, and basically what happens with contraction, you can see here is a microscopic picture of a relaxed muscle cell, this is on page 288, and then when they contract, you can see that the myosin has pulled the actin together. And you can see that also in the microscopic slide on page 288. Okay, we kind of powered through that because I wanted to see if we can get to heart. So next week we're going to do classification and function of muscles, but that was the skeletal muscle. We're going to switch gears now and go to the next lecture. The good thing is, this segment, this topic really does have heart. I say something like that, my wife just shakes her head and goes, the rest of my life. I'm stuck with him the rest of my life. There's a story, I don't know if anybody knows the evangelist Billy Graham, his wife when she was alive was being interviewed by Diane Sawyer, Diane Sawyer, and Diane Sawyer said, now Mrs. Graham, really, have you never ever thought of divorce? And Rosalind Graham leaned in and said, Diane, divorce never. Murder, however. He was kidding, we think. Okay, so this will basically be it in terms of the heart. Again, very, very basic on heart. But you know, I know people like get an update on this little website I get that there are people doing research on the pressure loop dynamics of a healthy heart versus a heart congestive failure heart versus a diabetic heart versus a transplant heart, and the biochemistry and the metabolism and all those kind of things. The heart is more complicated than what we're going to present today. Even if what we present at seems complicated, it's either because it's your initial exposure to it or your high school teacher didn't teach you anything. Just kidding. But basically, its primary function is it's a transport system, right? The function, what is a transport? Yeah, but what is blood? What is it? What is the heart transport? Blood, right? Consists of two side-by-side pumps. So the right side receives oxygen, poor blood from tissues, and it pumps the blood to the lungs, gets rid of carbon dioxide, and picks up oxygen. So, I mean, if you're going to draw it kind of schematically correct, you draw two small boxes with one really large box and one kind of smaller box. And then the right side receives it doesn't go to the ventricle. So the right side receives deoxygenated blood, sends it down to this chamber, which sends it to the lungs. And then the left side receives deoxygenated blood from the lungs, sends it down to the big chamber, the big dog, left ventricle, which sends it to the body, which goes back. That's basically your basic schematic of the heart. Like that's it. I'll tell you right now, if you want to learn the structures of the heart, and you do want to learn the structures of the heart that we're going to cover, the best way to do it is follow the path of the blood flow through the heart and list the structures as it passes them. Yeah. That's what we do. Works for me. So here, see from your textbook, page 664, you have kind of a fancier schematic of what's happening. Systemic circuit refers to the circulation within the body, and then the pulmonary circuit refers to the circulation within the lungs. And again, it's a closed system, but we're learning new things every day. For example, when you're exercising at extremely high intensities, if you're an aerobically trained athlete and you're exercising at extremely high intensities, your lungs, in order to deal with increased flow, increased pressure, will actually have shunts, which means it will let parts of the blood bypass the lungs, like a little safety valve, just so that it can deal with the pressure and the volume. So the receiving chambers are the atria, which are the top chambers, and atria like the atrium. So if you ever happen, guys and girls, to like meet someone who has a lot of money, they live out on Kempenfeld Bay, and you go into their home and the butler answers the door, and you step into that front room, that's the atrium. Oh yes, please wait at the atrium, we'll call this. Alright, so the atria is the receiving chamber, the ventricles are the pumping chambers. Those are the workhorses, especially the left ventricle. That really is the workhorse of your heart. That is what pumps all the blood to all of your body. This means all of your body. Okay. About the size of the fist, weighs about 1 pound. So everybody make a tight fist. Like you're going to a Wicked Wednesday tomorrow night, somebody insulted your mom and you're about to take him out. Okay? That's the average size of a heart. Now, loosen up your fist a little bit, just a little bit, like loosen up that if you were to punch up